Okay, hello, and today I will be presenting my NutriSci 623 final, and the topic that I've chosen for my set of papers, I chose the Selenium A papers. These is the biological one trick, multiple functions of a protein in different contexts. Now, a little bit of background. The point I'm really trying to prove here is that throughout these three papers, there are very general theme that I can see, which is the protein function across species are very similar in structure. The function can be, say, the same set of reactions, but depends on the context, they are very different, and the purposes are different. We're going to talk about a little bit about the evolutionary conservation or how the force of evolutionary plays between acquiring a function from another species, and also the process of recycling. And as recycling I'm talking about is that employing this one single protein in multiple places in the body. Now the outline, basically I have the three papers, and each paper here is going to prove a point. So paper number one, a virus carries a protein similar to human glutathione peroxidase, and this virus is this element that they're carrying is actually a eukaryotic element, which is the sexus element we're going to talk about. And number two, the protein PHGPX has different function in early and mature sperm development. And number three, a promiscuous transporter has preference ranking according to the need of the body. All right, so diving into the first paper, we have this virus, which is the MCV virus, and it seems that they carry a sexist element, which is very similar to the wild type human sexist element. And what it did here was that they took this element from the virus and they kind of transfect that to human cells. And it seems like the element that was transfected from the virus worked as well as the wild type. So this figure here proves us that the sexist element of a virus protein, they are interchangeable with the human protein. And next up, with the next figure, over here we have these protein that was encoded by this MC066L gene is functional very similar to human glutathione peroxidase. So P35 here is going to be your negative control, which is P35 is from another virus, is a apoptosis inhibitor protein, and the function of these of MC066L is that it seems to inhibit apoptosis via means of recovering from or protecting from free radical damage, which is very similar to the function of human glutathione peroxidase. And early on, we talked about the process of evolution, how one species gain another function from another species. It is very likely that the virus gained this function of being protected from apoptosis in order to infect human better or infect other species better under extreme conditions of sunlight and whatnot. All right, moving on to the next paper, we have these one single protein which is called PHGPX, and it seems like they have two different purposes in terms of development of the sperm. So in this figure here, what they did was that they extract the mitochondrial capsule of the mature sperm, and they found out that the majority of the protein in here has a lot of PHGPX, but despite the high concentration, the function shows to be mostly structural. It's not really enzymatically active at all. So why is that? We're just going to show on this next slide. So we have over here the same protein, but one is from mature, one's from the immature sperm. And in the mature sperm, we particularly want to look at lane 4 here. With the presence of H2O, H2O2, this is gonna cause a cross link, which make themselves aggregate or solidify in a sense, and this will help with the structural function of the sperm, whereas in the immature version, they were they were incubating the protein with GSH around, and with the presence of GSH, the protein is soluble, and as we can see, the protein is not really aggregating at all, and with the papers they cited, these proteins are also has a lot of enzymatic activity, which shows that in the mature form, in the mature version, it seems like this PHEPX mainly works as a structural support, whereas in the immature sperm, they want to protect the immature materials from being damaged under H2O2 exposure with the support of this GSH enzyme. Now, moving on to the last paper, which talks about this protein called Gen 1P. And Gen 1P has been shown to be a protein that helps with monocarboxylate uptake. But it turns out that Gen 1P is but also participate in selenite and lactate uptake. But however, these monocarboxylates, they effectively act as a competitive inhibitor to these to selenite and lactate. So when they add in these monocarboxylate, then the uptake of selenite and lactate would decrease, hinting that okay, there seems to be a preferential treatment of of binding monocarboxylate over selenite and lactate. So it seems like this one single enzyme can participate in transporting many things. And the structure of these selenite, lactate, and monocarboxylate are mimics of one another, so they're very similarly to each other, and which this enzyme can exploit this similar in structures to first they can bind all of them, but they can also utilize these small differences to prefer one over another, just based on what exactly the body is needing. So, in conclusion, a useful functional protein can be shared across species for different purposes. For here, an example of the virus and versus the human. The humans trying to protect from radical damage. The virus trying to do the same thing, but because they're infecting under the skin layer, they want to survive better. And one species can acquire from one another function. So, for example, the virus pick up that eukaryotic sexist element from a eukaryote, and these functions can be depend on species. So, either virus or humans, the location of the body. So it depends on where the protein is based. It can have a different function or a different purpose. A stage of life talk about this in the same protein PHGPX that have a protective function from radical damage in the early stage of of the sperm versus the structural function in the mature stage, and the body needs of energy where one single transporter can bind different substance, and they have a preferential ranking of these binding affinity. And here are the references. Thank you for listening.